Glycolysis. In aerobic and anaerobic respiration, the initial sequence of events are same. So they collectively called glycolysis. Thus, glycolysis is present in all type of living organism. So in glycolysis, one molecule of glucose broken down into two molecules of pyruvic acid. Now the entire process of glycolysis take place in cytoplasm. So this glycolysis, it doesn't involve or it doesn't require oxygen. Now glycolysis is also called EMP pathway. That means Emden, Meyerhoff and Parnas are the scientists. So they are actually discovered glycolysis. So this glycolysis is also called EMP pathway. So in glycolysis, it involves two phases. So the first phase is actually the activation phase or energy consuming phase. Second phase is actually the payoff phase or energy producing phase. So anyhow, so in the first step, see glucose is converted into glucose 6-phosphate in the presence of hexokinase enzyme. So in this process, one molecule of ATP it is converted into ADP, and the entire process it take place in the presence of Mg2 plus. Okay, so that keep in your mind, glucose converted into glucose 6-phosphate. Now this glucose 6-phosphate it is isomerized into fructose 6-phosphate. Okay, so now this fructose 6-phosphate it is converted into fructose 1,6-bisphosphate. So here in the first step, one phosphate group is added in the 6th carbon position, and the third step, phosphate group is added in the 1st carbon position. Okay, so here fructose 6-phosphate it is converted into fructose 1,6-bisphosphate. Here also one molecule of ATP is converted into ADP, and in the presence of Mg2 plus it is actually take place. Now fructose 1,6-bisphosphate it is actually in the presence of aldolase enzyme it is actually converted into triose phosphate. Okay, so here 6 molecules of fructose 1,6-bisphosphate converted into 3 molecules of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. Now see this glyceraldehyde 3-phosphate it can convert into dihydroxyacetone phosphate in the presence of isomerase enzyme, vice versa. Okay, now glyceraldehyde 3-phosphate, see from here onwards actually the second phase we will call it as energy producing phase will start. Now in the first phase already I told you that energy consuming phase. So in 2 steps ATPs are consumed. Okay, now here glyceraldehyde 3-phosphate it is converted into 1,3-bisphosphoglyceric acid. Okay, glyceraldehyde 3-phosphate 1,3-bisphosphoglyceric acid. See here this entire process will take place in the presence of glyceraldehyde 3-phosphate dehydrogenase enzyme. Okay, in the presence of glyceraldehyde 3-phosphate dehydrogenase enzyme, and here like see one NAD is converted into NADH plus. Okay, or NADH2. Okay, that you keep in your mind. Now 1,3-bisphosphoglyceric acid it is again converted into 3-phosphoglyceric acid. So here 2 molecules of ATP is actually produced. Why 2 molecules of ATP as like 2 molecules of NADH2? As I have already told you that see when fructose 1,6-bisphosphate is divided into 3 molecules of 3 carbon compound glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this glyceraldehyde 3-phosphate will be converted into pyruvic acid. At the same time dihydroxyacetone phosphate also can carry the same processes. Okay, that is why I'm just explaining that 2 molecules of NADH2 and 2 molecules of ATP is actually produced. Now here see here 1,3-bisphosphoglyceric acid it is converted into 3-phosphoglyceric acid. So here see this 1,3-bisphosphoglyceric acid when it is converted into 3-phosphoglyceric acid, as I have already told you that 2 ATP is produced. So it is actually take place in the presence of phosphoglycerate kinase enzyme and Mg2 plus. Okay, phosphoglycerate kinase enzyme and Mg2 plus. Now 3-phosphoglyceric acid it is actually converted into 2-phosphoglycerate. Okay, 3-phosphoglyceric acid converted into 2-phosphoglycerate in the presence of phosphoglyceromutase. So in the presence of phosphoglyceromutase, it is converted to 2-phosphoglycerate. Okay, now this 2-phosphoglycerate, it is again converted into phosphoenol pyruvate. Okay, phosphoenol pyruvate in the presence of analyze enzyme and NT2 plus. So here one molecules of water is also releases. Okay, now phosphoenol pyruvate, it is converted to pyruvic acid. So there pyruvate enzyme kinase is the enzyme, and at this time also ADP is converted into ATP. So two steps are there where ADP converted into ATP, and one step where NAD converted into NADH2. Okay, as I have already told you that fructose 1,6-bisphosphate, it is converted into glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. So this glyceraldehyde 3-phosphate, it can convert into pyruvic acid. At the same time, dihydroxy acetone phosphate can convert into glyceraldehyde 3-phosphate, and which will also convert into pyruvic acid. So here, so when a glycolysis, as I have already told you that two steps are there where ATP is actually utilized, and two steps are there where ATP is actually produces. So total of 4 ATP is actually produced, 2 are utilized, and 2 NADH2 is also produces.